Dr. Szynski, you're still muted. Please unmute
Yes.
yourself.
Now I'm unmuted, right?
Yes.
Yeah. So it's good afternoon, everybody. It gives me a great pleasure to welcome my good friend, Anindya Datta. Anindya got his PhD from Rockefeller University in New York, followed by postdoctoral training in Cold Spring Harbor Laboratory. And then he got his first faculty position here in Boston at the Department of Pathology at Brigham and Women's Hospital. And that's actually when we met. He then moved to University of Virginia, where he became department chair in the Department of Biochemistry and Molecular Genetics. And then quite recently, he moved to the Department of Genetics at the University of Alabama, where he is currently a professor and department chair. Anindya is responsible for several textbook discoveries and many things that we read that we take for granted about the DNA replication machinery we know thanks to work of Anindya. For example, he was responsible for cloning of ORC proteins of CDC6, for CDT1, and for MCMs. And quite recently, his lab made an amazing discovery that actually cell proliferation can take place without ORC proteins. These are just the examples of several really seminal discoveries in the field of DNA replication and cell cycle. Now, in addition, his lab has moved into different direction, studying non-coding RNAs in cell proliferation, in regulating cell proliferation and cell differentiation. Anindya got several awards and honors, and the list is so long that I will not read it because it will take too long. So I will just say that it's truly a great honor and pleasure for us to welcome you. I just also, as a housekeeping thing, I'm asked to remind everybody that you should post your questions in the Q&A tab, not in the chat. And with this, I welcome again Anindya, and Anindya, take it away.
Thank you very much, Peter, for the invitation and for that very kind introduction. It's absolutely a pleasure to be talking before such a, what should I say, collection of wonderful friends that we have seen for our, let me just, since I can't do two things at the same time, I'm first going to fix this. Yeah, for this opportunity to come and speak to so many good friends. I should start off by saying that I noted with regret that David Livingstone passed away a few weeks back, and my heart goes out to his many colleagues and his many mentees, some of whom might be in the audience. He was, as all of you know, was somebody whose presence, he was larger than life, with this big hearty laugh and incredibly intense and competitive, of course. I think Dana-Farber and the whole scientific world will miss him. So Peter, being a good friend for about 35 years now, more, pointed out that I have to give a talk that has to impress you guys. And knowing the clientele out there, I'm slightly nervous. So I decided to go through our repertoire of stories in the lab and picked out the weirdest ones. And they'll be mostly in the form of vignettes. I'll talk about the circles of DNA that I've been puzzling over in this because they're present even in normal cells. I'll talk about the small short RNAs that we discovered, which have taken off on a life of their own. And I'll talk about something that you would have ever expected to have done, but I'm actually doing old-fashioned GWAS studies and looking at the role of germline variants, and I'll tell you why I'm excited about it. So this opening slide also serves as a little bit of an advertisement. The name HereSync is here because University of Alabama at Birmingham got a naming gift, substantial naming gift. So there's a huge expansion in the research effort out here. The quality of people here are outstanding. Some of you even probably know people like Barry Sleckman. And since I'm the chair of a department that has to do quite a bit of building, I'm planning to recruit about 10 to 15 faculty members over the next few years. So if you have got very smart postdocs, if any of you are looking for a position, please drop me a line. some of the jobs have already been advertised. Now, knowing that people in Boston are just like the people in New York, in that the world is just Boston and New York, you then fly over country and then California, I thought I'll point out to where Alabama is relative to where I met most of you. So most of you I met in this times, Rockefeller, Cold Spring Harbor, Brigham and Women's Hospital. But then 18 years back, I moved to University of Virginia, and that was already kind of far away from Boston. And now after 18 years, I got the courage to move even further. And so here I am. So let's turn to the extrachromosomal circles of DNA. They are known as ECC-DNA, but I particularly like the word microDNA because the ones that I'm describing are these small circles that are present even in normal cells. And let me summarize what we know. So the conventional view of the genome is that the DNA is all entirely in these long chromosomes, the nuclear DNA. And what you're finding is that there's a reasonable repertoire of small circles of DNA present in normal cells. And not only are they present, but they're somatically mosaic in that different cells have different repertoire of circles. I think that's why they have been missed, because they don't contribute to copy number variations. In cancers, there are larger circles which then get selected for. So then they are no longer mosaic. Many of the tumors have those circles, and those were initially noticed as copy number variations. But these small circles present in normal cells are mosaic. So another thing that I want to address right away is that when the small circles got discovered, we initially thought that they're being produced by some kind of an excision process where this blue DNA is excised for the DNA, and that would leave microdeletions in the chromosomes. We do see microDNAs deletions in the chromosomes, but they are also somatically mosaic, and they're difficult to pick up. But the circles are very easy to see. So we are not sure whether all circles come from excision events like this, or whether circles can also emerge from copying events where there's an extra copy of this blue DNA which is forming the circles. And of course, as is likely in science, both these phenomena are probably happening at unstable sites in the genome. So the field has picked up. We sort of use genomics techniques to discover these circles by rolling circle amplification and high throughput sequencing. And then soon after that, they have been seen in Saccharomyces cerevisiae. We and others found them in other vertebrates. They've been found in C. elegans. But the maximum amount of attention has come from the circles in cancers, because using the same genomic techniques, people like these folks here have begun to discover that the circles are way more abundant than what was thought in the past. When initially the circles were seen just by cytogenetics and karyotyping, they thought, oh, well, maybe about 1 to 2% of cancers, the weirdos, have the circles. And now the realization is dawning that these circles are present in a much, much larger, some estimates say 50 to 60%, I think 100% of cancers, but we'll see where that goes. And the field is correspondingly, mostly from these papers, I guess, on the cancers, has acquired a lot of attention. They're getting their usual news and views, even some lay publications. And now this is something I always worry about. Now there are specialized meetings on circles. So it's probably going to be time for me to leave the field soon. So these are the hypotheses we have tested about the small circles in normal tissues. We know that they dysregulate gene expression. I'll give you a short picture showing that. We know that they're a byproduct of DNA repair. And we know that these circles are released to the plasma and can be used for liquid biopsy. What we'd like to test is whether somatic mosaicism of these small circles give cells in normal tissues competitive advantage, or even cancer cells competitive advantage. And we haven't yet got around to that. So these are the ways the circles are being picked up. So we have done rolling circle amplification on exonuclease digested DNA. So exonuclease digestion makes sure that all linear DNA fragments are chewed away, and then whatever is left is amplified by random hexamers and rolling circle amplification, and those products are sequenced. And in the sequence, we look specifically for the junction that is created by a linear piece of DNA coming together. The circles can be seen by electron microscopy, and I'll show you two other newer, relatively newer methods that we have been using lately to look at the circles. They've even been found by restriction of digestion to break the circles and sequencing. Andy Fire actually did a heroic experiment. They did cesium chloride gradient centrifugation for purifying plasmids, and they found circles from cells. But the oldest method was karyotyping. That's where the double minutes, which are the original long circles, millions of bases long, were noticed in the late 70s, maybe even a little bit earlier. And of course, when fish came around, metaphase fish went on to support their presence. So this is what the circles look like electron microscopically, and this is a summary of what we have found till now. So normal tissues like the brain, most of the circles are less than 2 kb. That's why we call them microDNAs. In cancers, actually, only about 60% of the circles are less than 2 kb. 40% are longer than 2 kb, ranging from few hundred kb, large enough to contain genes that will be amplified, to the ones that are 2 to 3 million bases long, which are, of course, the double minutes. So in the days when we used to think that double minutes were the only form of circles, people said, oh, they were present in about 1.4% of the cancers. But now we think nearly 100% of cancers has this whole repertoire circles, and probably 50 to 60% these circles of about 100 to 200 kb, long enough to contain a gene which could be amplified, but not necessarily long enough to be detected by karyotyping. In normal tissues, we haven't found a single tissue where we can't find it. We can even find it in the sperm. Complexity. So this number is off, actually. In our last vetting from the mouse brain, we have found about 500,000 unique sites in the circles. So a quick clarification. When we sequence these things, of course, they're mapped to repetitive sequences. But when they map to repetitive sequences, we are never sure could these be coming from tandem repetitive sequences, so we ignored them. So we only take into account the unique sites that are seen in the circles. And that's the number is not 10 to 40,000, is 500,000. And consequently, in cancer cell lines, this is going to get updated also to probably 500,000 sites. Remember, these are somatically mosaic, so every cell doesn't contain the same circle. I did have that update there. And the sites where these small circles are coming from are both from genic and intergenic areas, but there's a particular enrichment from epigenetically active areas. So places with activating chromatin marks, from CPG islands, from exons, from 5' UTRs. And there's a very nice peak that's symmetrically distributed within about 2 kb upstream of downstream of transcription start sites. So with these features, you can imagine, and that's absolutely true, there are hot spots in the chromosome. So all parts of the chromosomes don't produce the circles uniformly. There are hot spots that have these features. And as I said, there's somatically mosaic until there has been selection. If there's selection, then you start seeing them as copy number variation. So, do they regulate gene expression? For the long circles, there's never any doubt, because gene amplification in cancer, some of you might even remember that MDM2, the famous oncogene, was identified as a gene present on mouse double minute, MDM. It was a second gene on the mouse double minute. So that has been known for now over 40 years. But what are the short circles? So here, Teresa here did this interesting experiment. She noticed that in many of the circles that we are pulling out and sequencing, they included the microRNA gene. So as you know, the microRNA gene comes as part of a precursor microRNA. So it's very small. It's maybe about 50 bases long, maybe 70 bases if you're generous, which is usually in part of an intron. So the promoter for the primary microRNA is 20 to 40 kilobases away. So obviously these circles being 100 to 200 bases long, sometimes about 400 bases long, only contain the precursor microRNA gene. So you wondered, if you produced a circle like that, would it express the precursor microRNA? And the answer was yes. Would it express the microRNA? Yes. And would it repress genes? And the answer was yes to all of them. All of this is happening without any apparent promoter on the circle. So we think that either the torsional stress in the circle or the dechromatinization on the circle accounts for spurious promoters springing up, and that's how this gene is being expressed. Teresa went on to do another very cool experiment. She noticed that some circles were coming from within exons, and she wondered whether that would produce short shRNAs or siRNAs from transcripts from both strands, and whether that would silence the parental gene, and that's exactly what she found. Now, these, of course, say that this can happen. We still haven't been able to show that this does happen and that the cell actually takes advantage, or the organism takes advantage of it, or a disease takes advantage of it. And that's where our quest lies. But to introduce a technique that we're using now, I'll start off by saying that the question we wanted to know is what produces these circles. And the problem was, firstly, sequencing is expensive. Secondly, the sequencing approaches, because they use a rolling circle amplification, we lose a lot of the quantitative data. So sequencing is great for finding the complexity of the library, how many sites are producing circles. But for abundance, we needed a cleaner and simpler assay. And to develop this, Teresa adapted inverse PCR, which many of us are familiar with. The idea was that if this red sequence comes out and forms a circle, if she produces designs outward directed primers and tries to do a PCR, there wouldn't be any product on genomic DNA. But if this thing got circularized, there would be products. You would say, aha, but direct repeats of this sequence on the chromosome would also give you products. So the precaution you take is that after you try to harvest fractions of the cells that are rich in the circles, but worried about broken DNA fragments, you then first use exonucleases to digest all broken DNA fragments and then do this inverse PCR. It turned out to be a fairly good assay, though slightly noisy. So we never examine just one site in the genome. We examine a basket of sites, usually about 8 to 10 sites, and we always have a gazillion replicates. And then we try to get an idea about whether a particular manipulation of the cell is changing the abundance of the circles. So the first big hit we got was the double-stranded breaks of any type, whether it's an X-ray, as you can see here, bleomycin or NCS, they all increase the formation of these circles. So you can see it's about a twofold increase, 2 to 2.5-fold increase, and within about 48 hours of treatment with these double-strand break-inducing agents. But here, we are worried that the double-strand breaks came close together, released a DNA fragment, and that's what got circularized, and that's what we are looking at. So we tested whether a single break directed by a CRISPR break would be enough to form circles from the neighborhood of the break. And much to our surprise, that's exactly what we find. So here's a CRISPR-Cas9 cut, and we put these outward-directed primers up to about 2 kb, and we could see, again, within 48 hours of introducing the cut, an increase in this yield of circles from this neighborhood. We don't see the circles this increase more than 2 kb away. That's sort of the limit within which we can see it. We also don't see the presence of the circles from other sites of the genome increasing when we introduce a very specific CRISPR-Cas9 cut. So we think it's related to something that's happening locally after the introduction of the cut. What could that be? So obviously, the thing that we wanted to turn to was break DNA repair pathways, but we already had some strange hints by screening these chicken DT40 cells where there had been mutations in mutations engineered in multiple DNA repair genes. And in the olden days, when we were trying to measure the abundance of circles by EM, we had noticed that this mismatch repair mutant DT40 has about a 10-fold or 5-fold decrease in abundance of circles. We reproduced this result by inverse PCR, not only in DT40, but also in mammalian cells. And we now know by inverse PCR that defects in mismatch repair, defects in genes that are in very early stage of recognition of double-strand break, like NBS1, they decrease the abundance of the circles. And then there's this nuclease, which initially were thought to be involved in Fanconi anemia repair, but nowadays is not believed to be. This nuclease seems to be also important for the formation of circles. So once we realized that this assay was working, we went and screened a whole lot of repair pathways. So if you remember, when there's a double-strand break, the repair can first happen without any resection, where the strands are just brought together and stuck together. That's classic non-homologous end joining. Or alternatively, there could be a little resection or a significant resection, and then the microhomologies in this case, single strand bits in this case, are annealed together to form, to repair the break. And so that's MMEJ and single strand annealing. And if all else fails, the resected strand will, and if a daughter strand is present around, mostly in late SNG2, the resected strand will invade the sister DNA and will repair the break by homologous recombination, right? So what we're finding is pretty interesting. The things that decrease circle formation are all in this arm, these red boxes. They decrease circle formation when they're mutated, suggesting that these pathways in some manner help in circle formation. The weird one here is the mismatch repair pathway. We still haven't quite figured out unless it is involved in this MMEJ process to get rid of mismatches. It may be. We don't quite know how to test whether this involvement is dependent on this. But the strange result was here. Mutations in classic NHEJ pathways actually increase circle formation. So all of these mutations or chemical inhibitors were increasing circle formation. So what that suggests is that when there's a double strand break, if there's no resection, then the stitching together of the DNA strands can happen without circle formation. But if there is resection, then the cell has problems and ends up creating these circles. We don't have a great hypothesis, but the best I can think of is, since it can also arise from a single break, is that the single strand DNA, if it's sufficiently long, can get into trouble by getting microhomologies that end up annealing to other parts of the DNA. And to get rid of this, the cell sometimes uses the expedited method of just excising and ligating these ends and thus forming a circle. Now, there's DNA lost in this case. So in this case, there might be microdeletions, but it's also possible that since there's another copy of the single strand DNA in the other end, it's possible that that could be repaired before that DNA could be recovered before the repair is completed. We suspect that's why microhomology mediated proteins are involved here. So it's not classic MMEJ, but the use of the proteins for a different pathway. But we'll keep you posted as things develop. Now let's turn to cancers. So judging by the experience of the many people who are studying the intermediate to long circles in cancers, we started wondering whether we could detect these circles in cancers even before a gene has been amplified. That would be pretty cool, actually. If you could find that a cancer has got a few cells which have these circles and which would then take over the population in the face of a particular type of therapy, we might be able to change the therapy. So we're trying to develop a new method to identify them. And this is one I keep on talking about and still haven't done much with it, which is that does identifying these circulating circles, especially the longer circles coming out for cancers, would that be advantageous for liquid biopsy? And then there's, of course, the eternal question, why do cancers make these much longer circles? And also, how do they survive? Like, why don't they get lost in each cell cycle? And no one really has a clue on any of this right now. There are hypotheses about how the long circles are formed. I'll touch on that in a second. So here's one of the hypotheses, which is that my friend David Pellman's discovery that micronuclei leads to breakdown of DNA into multiple fragments and then subsequent joining of these fragments to form chromotryptic chromosomes was adapted to suggest that the same kind of joining might form a multilocular large circle. And that's probably happening. And some of you might know Fred All's recent paper where he said that post-apoptosis, these long multilocular circles can get formed and get released and can stimulate the immune system. So that's probably the mode of origin of the long circles in cancers. But the single locus small circles that we're seeing in normal tissues are probably happening by some kind of anomaly during repair or during replication stress, which also will produce single-stranded DNA breaks. So back to the question of pre-amplification. Can we detect a cell which has, say, an EGFR gene, if you're looking for the circles, before that circle has taken over the whole population of cancers? Once it's taken over the whole population, it's of course very easy to find that circle by copy number increase. And to address this, we decided to go through existing data. We wanted some techniques that are already being used by everyone. Nobody does rolling circle amplification, right? As a general cancer, in TCGA or something like that. But people do ATAC-seq. And what Pankaj in the lab discovered is that in ATAC-seq, the tagmentation can also break open circles. So in a small proportion of the cases, the sequencing from the ATAC-seq will pick up this ligation junction on the basis of which the circle is identified. Now, again, people will say, well, yes, but you also will pick up direct repeats. And the answer is yes, you can. I would ideally like to do tagmentation, which you can do in cells in culture, but no one has done it in actual cancers after exonuclease digestion to get rid of linear DNA, but that hasn't happened yet. So let's first see if this strategy with cells in culture, where we can do the exonuclease step to remove linear DNA, helps. The answer was yes. We could detect hundreds of thousands of circles in these two cancer cell lines. And here you can see this interesting point. If you take this 1 kb cutoff or 2 kb cutoff, there's a 40% of the circles are longer than that 1 or 2 kb microDNA limit. So you can also pick up the longer circles, which will of course be unilocular here, because we are identifying the breakpoint only if you know the DNA segments that are being brought together. The multilocular circles are still very hard to pick out by this junction method, because they'll just look like translocations, right? Fragment A is ligated to fragment B, C, D. Eventually, fragment E might circle back to A, but it's hard to map things around the circle. So we found even these longer circles discovered by ATAC-seq to be present all over the genome in cancer cell lines, and we validated 11 out of 11 of them by inverse PCR. We even validated them by metaphase FISH. So you can see the circle here on the metaphase FISH, which is off the blue chromosomes. So now we could turn to TCGA data and do the experiment to see whether in ATAC-seq data, there's a possibility of finding circles even before they have taken over the population. And indeed, there were at least 2 patients where we found, at least 1 patient where we found that this EGFR gene was present as a circle. By copy number amplification, this increase, this step up is not seen in this patient. So we are picking up the circle in this patient even before the tumor has selected for the circle. And I'm fascinated by the possibility that this could tell us, if you are to treat this patient, for example, with anti-EGFR therapy, we better be careful that subsequent amplification doesn't make the tumor resistant. So this is all that I've told you. I won't read it all again. I'm particularly interested in finding how these small circles, where they contribute to normal physiology, and regarding the long circles, how they're sustained and maintained. The folks who did the lab, the original discovery of the small circles in the normal tissues was done by this bioinformatician and this very talented molecular biologist, Yoshi Shibata, Pankaj Kumar, and they've subsequently, the project has had work from Dr. Laura Dillon, now Dr. Teresa Paulson, and Zhangli Su. And we have had a lot of collaborators all over and appreciate their help. All right, I know that the questions and answers will come by electronically, so I'm just going to charge ahead. So let's talk about another strange molecule discovered from genomics in the lab. These are these tRNA-derived RNA fragments, or TRFs. I'm going to try to convince you that in this family, this repertoire of small RNAs, it's time to add TRFs to this. So how did you discover them? This was in 2006, we were using like high throughput, but the other method whose name I'm going to forget, where you could get about 500,000 reads. And we found nearly half the, 2/3 of the reads were not mapping back to microRNA genes, known microRNA genes. What was worse, they're not even mapping back to the hairpin that's characteristic of the precursor microRNA. So where are they coming from? We noticed about half of them were coming from tRNAs, at which point worry was that there are degradation fragments. But then we noticed a very peculiar trend about them. So tRNA start off life as a precursor tRNA, the leader and the trailer is clipped off, the CCA gets added, and that's the mature tRNA that you are familiar with that folds into that cloverleaf pattern. Now, if these were degradation products, you'd imagine that they'd come from all tRNA loci, they'd be distributed all across the length of the locus, but that's not what we saw. Instead, we usually see from one mature tRNA, maybe one of these guys, just a tRNA fragment coming from the extreme 5' end, a TRF5. You don't see the others. On the other hand, from another mature tRNA, you might see a TRF3, but nothing else. And then there'll be a few others where you just see a TRF1 from the trailer sequence and nothing other. In other words, there's some specificity. What's more, we did a meta-analysis of, I don't know, at that time there were probably 60 to 100 microRNA libraries, and everybody's libraries had the same fragments, the same sets of fragments. So it wasn't just a random assortment of fragments being picked up by different people. And of course, everybody was ignoring them. But now, actually, this number is now over 1,000 papers. They have acquired different names. They're called TRFs. They're called tRNA-derived short RNAs, TIRs, short RNAs, tRNA halves. They're very specific fragment that's saved. They're seen in all tissues that people have looked at, all the way down to the yeasts and even some bacteria. Very early on, using dicer knockout or Drosha knockout cells and looking at the small RNA-seq, we could clearly show that these are dicer independent and Drosha independent. So then the question becomes, oh, then they probably don't associate with AGO. But again, as we surveyed AGO par clip data, it was very clear to see that not only these short fragments associate with AGO, but they also bring to the AGO complex target messenger RNAs that are recognized by complementarity to the seed sequence. So all of this was well and good. The real question, let me just give you a summary of where things stand. So these are all the TRF5s coming from the 5' end. These are the TRF3s. There's two different species that show up, which will become important in some subsequent work that I won't describe to you here. And then there are the tRNA halves created by a cut in the anticodon loop. And there are even some internal fragments, but not as many. And they're much rarer than the 5s and 3s. But what you ask is, come on, show me some experiment. Show me that TRFs really regulate gene expression. And this very neat experiment was done by Janan Kuscu, where she overexpressed a tRNA gene and showed that overexpression of the tRNA increased the level of the TRF3. And so the question she then asked is, if you have a luciferase at the 3' UTR that has got the complementary sequence for the TRF3, so it's the target site, will it be repressed? So let's look at it. And the answer is, of course, yes. So these are the different targets. Here's a target without any complementary site, a target that's complementary to TRF3-3, another that's complementary to 3-9, another that's complementary to 3-1. So now if the tRNA expresses just 3-3, doesn't express the other TRFs, you'll see that it will repress the complementary target, but not the non-complementary targets, right? So there's specificity in this repression. And you can see that you can change the specificity by changing which tRNA you're overexpressing. So when you overexpress this guy, it will inhibit its cognate target, but none of the others. So it is specific. So using this assay in this paper, we could again show that the TRF production and luciferase repression does not require dicer. It requires the Argonaut. This is a little bit controversial because there's at least in Drosophila, there was a suggestion that dicer has a role in feeding the short RNAs into Argonaut. But actually in mammalian cells, that's been questioned already. And I think this kind of lays that whole issue to rest, that these guys are being produced by a dicer independent method, but they still enter into the Argonaut. So we could make mutations in the target seed sequence and showed that the target has to match with the seed sequence of the TRF and not as much with the rest of the TRF to be repressed, and that these TRF3s repress endogenous genes when they're overexpressed. Great. So where are we now? So of course, we want to show that these things are relevant in some disease or something. And so we turn to cancers. So the first worry was, are these sufficiently abundant? So we surveyed lower grade gliomas in TCGA, and we first measured the microRNA abundance along in all the tumors that you see. And nearly 80% of the microRNAs that have been indexed are below this level of detection, right? This is the median abundance. There are very few that are at this levels of abundance. Now, the microRNAs that have been implicated in LGGs, they affect outcome, they affect certain cellular phenotypes, are shown off here, 124, 15, 155. So that's sort of the physiological abundance level at which microRNAs have an effect. So here are 3 TRFs. 3-30, 3-21, 3-9 that have comparable abundance, and they have very nice effect on TCGA on outcome. All three higher levels of expression predict poor outcome. Now, whenever you see data like that, you always feel queasy. Could it be just because the tRNA levels were high, and that's why the TRF is high, and that's why it's have those tumors, those patients do poorly? But then one of them was particularly interesting because this 3-30, because there's a clear difference between the prognostic effect in males versus females. In males, it's very prognostic. In females, they're not. So it can't be because whatever is responsible for the increase in the level of the TRF is non-specifically bad for the patient, because there's some interaction with what the patient is, patient's composition, which makes me suspect that there's some kind of specificity going on. So another thing that some of you might remember, MYC is famous for turning on tRNAs. So you wondered whether MYC increases TRFs, and sure enough, taking these cells from very well rich whatever lab at MIT, Rick Young's lab at MIT, we could show that induction of MYC caused an induction of the TRFs. And then when you look at the same MYC-induced cells and look at the TRF targets versus the non-targets, the targets are all repressed relative to the non-targets. So something is happening that we don't quite understand. And then if you look at these TRFs, we sort of overexpressed a TRF in glioma cells, and we could see their target genes were repressed, and then a mismatched TRF did not repress, right, in several of the cases. And these all have target sites in them. And for all of them, if you remove the TRF, so now you're no longer overexpressing, because that's something that people start getting queasy about. So you're just removing endogenous TRF by an antisense oligo, and you can see that several of the targets are getting de-repressed. So it's making me think that the TRFs do regulate gene expression under normal circumstances. Do they change the phenotype of the cells? And this result is pretty intriguing. In astrocytes and even in glioma cells, we overexpress what I call these oncogenic TRFs, the ones whose expression predicts poor outcome, and they all stimulate invasive activity. So we are now trying to connect this phenotype to the gene repression and see if they're related or not. So this is all unpublished. So it's telling you that endogenous TRFs are expressed at levels sufficient to repress gene expression, and that high levels of at least 3 TRFs predict poor outcome. They're required for glioma cell proliferation. I didn't show you that, but they are by antisense experiments, and that these 3 TRFs stimulate glial cells invasion. So we'll keep you posted how that goes. And here, the hero of the work again is Young-Sun Lee, who discovered it originally, and is now in Seoul. Pankaj helped in the bioinformatics, and then with Manjari, and then Janan Kushchu did the very neat experiments with tRNA overexpression that showed that it's functional, that it can repress genes. And now Zhangli Su has taken it over and is trying to determine if the TRFs are affecting phenotypes in cancers. Right, the last part. This is completely outside anything my lab has ever done, but it again tells you how amazing these genomic strategies are for getting into new areas and for exploring things. So I'm going to tell you that germline variants have a significant effect on cancer outcome. And the insight was this. You probably have now read a gazillion papers that say that 2 patients come, similar cancer, similar grade, similar stage, but one person does well and responds to therapy and the other person doesn't. And to explain that difference, the focus has been on the somatic mutations in the tumor, because of course there are somatic mutations in the tumors. But the surprise is that there may be about a few hundred somatic mutations that differentiate these 2 tumors. There's a much larger repertoire of germline mutations, inherited mutations that are present in these cells and not in the others, and vice versa. Tens of thousands of germline variants distinguish individual people. Now, of course, if a germline variant can have additional effects on a cancer, not only by affecting the cancer cells' behavior, but by also affecting the microenvironment. angiogenesis, immune cell reaction. So you wondered whether we could look at it, because almost all work in cancers has been done with somatic mutations. And when people have looked at germline mutations, they have generally looked at it in the context of risk. There are famous examples, BRCA1 mutation, for example, right? And then once something has been implicated in risk, then people look at it also in outcome. And they say, yeah, there are variants that also affect outcome. The question we wanted to ask, just by a broad GWAS screen, can one first identify germline variants that will be predictive of patients' outcomes? So all patients have been diagnosed, so we are no longer looking at risk. And TCG is perfect for that. There are a few hundred patients. They're mostly in the United States. You have their grade, stage, and all these other variables. And the treatment is approximately standardized. And their outcome is recorded. So we decided to explore that. So Ajay, the MD PhD student who did, first developed his methods in gliomas. I won't take you through all the details. We initially focused just on the normal DNA that was present from the patients, but it dawned on us that we should also look at tumor DNA and tumor RNA and find mutations, but then use filters to remove somatic mutations and filters that remove RNA editing sites. And I can discuss how we did that, or you can go and look at the paper. And the other thing we're concerned about is that we could be overcalling sequence variants when they were read just a few times. So we needed a sequencing depth threshold. And then we introduced the biggest filter of all. We decided only to look at those variants that are accepted germline variants in the population. So the minor allele frequency in the human population had to be at least 5%. Now remember, this selection, we are probably throwing away the germline variants that have the highest effect, because the rarer variants are expected to have higher effects. That's sort of a general principle of genetics. But regardless, we thought we'll just take the ones that have already been reported in normal populations. So it's very unlikely that any of these changes coming through would happen at exactly the same base, at exactly the same residue, if they were already present in the population. And anyway, most of the ones that we pulled from here are confirmed from the sequencing reads in the normal. So we are pretty sure, and we did all the quality controls to know that these variants we're looking at are definitely bona fide germline variants. Then we do an old-fashioned Cox regression, but here, thanks to TCGA, we can put in all these other factors that are known to affect outcome. So for example, histological type, histological grade, IDH mutation, of course, Bill Kalin at Dana-Farber did a huge amount of work on that, other molecular markers, and so on. So there are too many clinical covariates, so we diminished them to about 11 by lasso regularization, but with that, it was a manageable Cox regression. And then we looked at what would the p-values look like. So these are all those 200,000 variants. And because we are looking at 200,000 variants, we had to look at an adjusted p-value to diminish the penalty for multiple hypothesis testing. So at an FDR of 0.1, 2 of these variants survived this screen. They're predicting poor outcome. So of course, we immediately focused in on this one because GRAB2 is well known to be in the RAS pathway, and it was in the 3' UTR. It turns out to be a variant that's comes from the Norwegian population. So this is what the hazard ratio looked like. So remember, I told you the minimal MAF is about 5% in the TCGA population. There are 21 heterozygotes. We didn't find any homozygotes in that initial search. There are actually now homozygotes that have been added to the TCGA. The interesting point was the hazard ratio is ridiculously high. I think this is a little bit of an artifact because there's so few heterozygotes, but it was a very significant hazard ratio difference. And this is the constructed Kaplan-Meier plots. This is the homozygous for common allele. This is heterozygous, right? And as I said, we didn't get homozygous for minor allele in this initial study. So now the question comes, is this something that's useful? So we used a standard ROC curve. And for those of you who are not familiar with that, remember that if it's random noise, the curve will go through this diagonal. If it's a perfect predictor, it will hug this corner. So this dark black line looks at what the ROC curve would look like using all the clinical information that we currently use. And this light gray line is what happens if you use the same clinical information and add the status of this RS617 in the GRAB2. there's a significant improvement in predictive ability. So this was really useful and interesting. And it made us wonder whether this shouldn't be something that should be adopted more often. So then the question evolved into, can we use TCGA to figure out whether this guy is even affecting the RAS pathway, or whether it's a linked variant somewhere else? Like when you pick up one variant, that may not be the causative variant. It could be other linked variants that are present in that same haplo block. So I remind you where GRAB2 is in the RAS pathway. And right at the end of the RAS pathway, there are known RAS target genes. So we first wanted to know whether the tumors with this variant, whether they have a differential expression of the RAS target genes. And we're really surprised. In a gene set enrichment analysis, you can see that the tumors that had the variant were actually expressing higher levels of the RAS target genes. So it seemed to be activating the RAS pathway. Again, may be true, may not be true. But at this point, we are, again, because in TCG all these data is available, we also started wondering whether this gene, which is a repressor for RAS target genes, and which is known to be mutated in subsets of glioma, whether there's a selection for this mutation in patients with this variant or not. It happens to be in the 19q interval, which is often deleted in gliomas. So the tumors are annotated as either having a 19q deletion or a Capicua mutation. And this is the result. In the patients who have the AA allele, which is the common allele, 15% of them have Capicua mutation, or 25% have the 1p19q deletion. In the ones that have the GRAB2, that variant, the numbers double, and the p-values are fairly significant. So there's something happening. The germline variant is either selecting for the somatic mutation, or under the influence of the germline variant, the somatic mutation appears in some manner. I doubt that second is true. I think there's some selection variation, but it might explain why the RAS pathway is turned on. So we started getting ambitious and said, well, I hate paying that such a high penalty for multiple hypothesis testing, because most of these variants, the numbers are not very large of patients with the variants. And so the study is not sufficiently powered. We actually haven't done that filter yet. But could we decrease the number of genes that is being screened by some other approach? And we decided to do a very simplistic minded approach, which is that we look at only those prognostic germline variants that change amino acid sequence, right? Remember, it's basically a biology lab. So we want to find the ones that are easy to study. So if the variant changes the amino acid sequence, we'll just look at those. So use that filter. Now there are only a few variants, germline variants like that, and most of them fall below the threshold, but a new variant with a much lower hazard ratio now reaches the FDR threshold. So what's this guy? So this one is in a gene called ANKDD1A. And this is the alternate allele, and it's present fairly frequently, 30% of the population. And in TCGA, you can see that the hazard ratio of having one copy of this allele was about 1.73. Now, fortunately, this allele is present in the Chinese population. In China, they're developing a similar glioma data set for CGGA. And so we decided to look at CGGA, and there again, we found the same hazard ratio. This is cool because you probably know this. GWAS studies are notorious for not reproducing in different populations, in different studies. But here, even before our publication, we could kind of reproduce it in the Chinese population. So this is probably a real finding. So this is ANKDD1A. This is the very highly conserved amino acid that's mutated to glutamic acid. See how well conserved it is. You have to figure out what it's doing, but it's telling you which way we are headed because we are finding all these variants. So Ajay, once he got a taste of that, got really excited and said he's going to look at all 33 cancers in TCG. And he went and did it. And it was pretty complicated, but eventually he's come up with about 180 odd prognostic germline variants. And I'm almost sure that this is the tip of the iceberg, that as people develop better and better patient populations, especially the rarer variants, which are likely to have higher effect sizes, you're probably going to get more and more of them. So this is what it looks like. Now, each of those plots, this is one cancer, ovarian cancer, and the red dots are the ones that cross the FDR threshold, right, in an analysis like this. So, I won't belabor the point. We can find variants that we can find in one cancer, and then once we have that variant and go and test it in another cancer, we find that that's predictive also. So for example, there are 5 such variants that are predictive in 7 or 8 cancer types. This has got to be real, excepting it might not be this variant, but could be a linked variant that's causing this difference in outcome. These are what the Kaplan-Meier plots look like. They're beautiful, actually. And this is the real Kaplan-Meier plots. This is not a constructed. So for example, one of my favorites is this one in rectal adenocarcinoma. This is the common allele. This is the heterozygous, and this is the homozygote for the minor allele. And you can see each of them have a different survival outcome. So we did all the other things, like many of them help the clinical prediction of the outcome. Many of these variants are in genes involved in cell death, cellular instability, genome instability, invasion, and metastasis, but many are unknown. And of course, those are the ones that particularly take my attention. Frankly, there's no way that my lab can do all of this. So I'm essentially publicizing this in the hope that some smart postdocs or graduate students will take them up and develop their labs around some of these variants. But of course, the most interesting thing that comes out of it is the mechanism by which they're acting. So we have done similar data analysis about like which genes are being affected, and some of them could be EQTLs that affect gene expression, and some of them could be affecting something that we don't really understand. Probably the most important thing is to validate it in a different population. Without that, yeah, this is interesting, but maybe not reproducible. We'll see. But the most practical application would be if one can make polygenic scores and can use that to improve predictability in cancers. So patients have been diagnosed and you're trying to decide how they will do. And even better, if you can predict which type of therapy they'll respond to, but that's still some way off. And I'm still interested in biology. At least if I can take 1 or 2 of these variants and explain how they're affecting the outcome, I'd be thrilled. And here, the hero of it is Ajay Chatra, an MD PhD student who dragged me into this area and did an incredible job. And he was assisted by these 2 bioinformaticians, Manjari and Pankaj, this really excellent cancer geneticist, statistical geneticist, Akrosh Ratan, and this is a statistician, because now we are in territory that's terribly scary for me. So we have to have a statistician here on our team. The other thing I wanted to advertise is that I am looking for postdocs. And so if anybody wants to join my lab, drop me a line. And that's the final conclusions. I'm going to leave that out. I've told you about these circles. I really want to use them for cancer management and or find that they have a function in normal tissues. We are now looking at how the TRFs affect physiology or pathology. We'll keep you posted how that happens. And these germline variants, probably the most practical approach is, can we use it to improve the management of patients? And the biological approach is, does it tell us something more about these cancers than we had expected till now? Thank you for your attention.
Yeah, thank you, Aninja, for a really fantastic and truly impressive talk. And then we have already one question, but I, of course, encourage people to write more. And the first question is from Johannes Walter.
Oh,
and
no, not Johannes.
No, Johannes says, hi, Aninja, great talk. I like your model that intramolecular Pol theta dependent annealing and extension creates the ECC DNAs. Could you test this further by targeting breaks to chromosomal sites where there are different levels of microhomology near the break and measure the level and size of the circles?
Wonderful idea. Really nice. Didn't even think about it. I have to write it down, Johannes, before I forget, but clearly you and I have to chat. Circles at, yeah, very good idea. Haven't done it, obviously. But if we do it, I'll let you know. But we should really talk anyway, because we are now totally into your area of expertise. So let's make a Zoom appointment next week. Great to hear from you.
Okay, unfortunately, he cannot answer you. He can only write and I can read it for you. So I have actually a question. If I understood correctly, I was really amazed that if there is a TRF that correlates with the survival in glioblastoma in one gender, but not another. Could you speculate that this is kind of mind-blowing?
Well, actually, that's known for several genes. You can actually look at certain genes. So most likely, it's interacting with the hormonal pathways, right? So most likely, we haven't done anything. This is like, this is one of the things about genomics, which is it's all correlation. It's not causative. So we don't know. But most likely, it's suggesting that there is something happening with hormone expressed or hormone regulated genes in the two sets of gliomas, and the TRFs affect them differentially.
Okay, thank you. Since I don't see more questions in the Q&A, while I'm waiting for questions, I'll ask more. You mentioned about, so I was really interested about those DNA circles, which I heard from you before. And then you mentioned in the introduction that this is present in normal tissues. Are they tissue specific?
So there's a weak tissue specificity, which is probably explained by tissue specific gene expression. So what do I mean by that? If you do principal components analysis of the sites where we are finding the circles from in different tissues, you can separate the tissues from one versus the other. It's no problem. But if you try to do hierarchical clustering and see if all the circle libraries that we've generated from a given tissue are closer together than others, there are occasional libraries that don't quite match. Now, if you do the same thing with RNA-seq, they match perfectly, right? You can easily separate different tissues by RNA-seq hierarchical clustering. So what it's telling us is that the circles, probably because they're dictated by where genes are being expressed, have some responsiveness to tissue of origin, but not as absolute as gene expression. It's going to be an important question for if you had ever to use it for liquid biopsy, right? Which tissue did the circles come from that increased in the blood?
Okay, there's another question from David P. Can you speculate about the role of MMR in ecDNA formation? Pellman, yeah, of course it's Pellman, I guess.
Yeah, I was wondering if it wasn't Pellman. So David, so I'm speculating, but you probably know that when there is annealing of two strands, whether it's microhomology mediated or even HR mediated, if there are mismatches, that attracts the mismatch repair complex to fix it before it can proceed further. This has been best demonstrated in the HR pathway. So I am imagining that's what we are seeing. But surprises might exist that we haven't yet spotted. So basically, the annealing happens and then there's mismatches. And because there are mismatches, the mismatch complex gets involved to fix it before the next step can come through.
Okay, thank you.
And if you disagree with that, I'd love to have a chat with you. I'm going to meet you afterwards anyway, if you have a better hypothesis.
Okay, while I'm waiting for more questions to come up, I will ask one more question. Did I understand correctly that you mentioned that that there is a certain degree of mosaicism in terms of those circles, but the cancers are not mosaic, they're more homogeneous?
Okay, so I didn't clarify that. In cancers, the focus has been on the long circles, and then they lose the mosaicism, right? Because the long circles often get selected and the mosaicism is lost. What I'm trying to say is that hidden behind those selected circles, there's also a lot of somatic mosaicism of the smaller circles, which nobody has paid attention to. Do they contribute to the circle? Do they contribute to the genomic instability of cancers, right? So that's what I was trying to say. I think selection is very important. If selection happens, then we realize that this is biologically significant. But without selection, if you're just looking at mosaicism, you're often stuck with, is this just noise, with no function? So I'm looking for a small circle that is getting selected in a cancer. Then I'll be very, very happy.
Yeah, thank you, Ninja. So there's a follow-up question to, not to this question, but for the previous question from Johannes Walter, who says, but then MMR would suppress ecDNA formation, no?
Yes, Johannes, you're right. Unless whatever is the next step to form the circle, unlike in HR where MMR suppresses HR elongation, unless the next step needs resolution of that mismatch. Might be relevant for microhomologies, because the microhomology has to be sufficiently stable for whatever is the next step that's going to happen. Complete speculation. I just have the observation, and I'm saying maybe it's the mismatch that's likely to happen when microhomologies are brought together that is responsible for the role of the mismatch repair. And it will depend on whether that mismatch pathway has to finish its job before it can go on to the next step. Again, total theory.
Great. I see no questions. Anindya, thank you so much for really fantastic talk and for really impressive covering of 3 different areas. Thank you very much. And then I understand you are meeting more faculty afterwards, right? So thank you everybody who joined us to listen to this fantastic talk. And then I'm leaving you, I guess, with David Spellman, right?
No, I think I'm supposed to log into a separate link.
Okay.
Thank you, Peter, for inviting me and hosting me. And thank you, Claudia, for organizing the whole thing. It was beautiful.
Thank you, Claudia. Thank you so much. Thank you.
Bye-bye.
Bye.